everyone, today we will discuss on module entitled Biopreservation of Food. The main objective of this module is to provide practical guidance on agents used in biopreservation of various food. Biopreservation can be defined as the extension of shelf life and enhanced safety of food by the use of antagonistic microorganisms and or their antimicrobial products. Biopreservation may consist of any of these approaches by adding bacterial strains that grow rapidly or produce antagonistic substances, by adding purified antagonistic substances, by adding the fermentation liquor or concentrate from antagonistic organisms, or by adding mesophilic lactic acid bacteria as a fail-safe protection against temperature abuse. Antagonism refers to the action of any organism that suppresses or interfere the normal growth and activity of any food pathogen or spoiler, such as various types of bacteria or fungi. Various microbial species have developed a diverse mode of mechanism of inhibiting or reducing the growth of other microorganisms through indirect mode, means by changing pH, osmotic pressure and surface tension, or direct mode, means nutrient and space competition by producing toxic component, antimicrobial component, bacteriocins, antibiotics, etc. Lactic acid bacteria are a group of bacteria which have been exploited since long for the production of fermented food due to their ability to produce desirable changes in the taste, flavor and texture along with inhibition of spoilage and pathogenic microorganisms. Thus, the most of bacteria from this group are known to be safer for use in food since long as so have GRAS status. Lactic acid bacteria are gram-positive, non-spore forming, non-motile, rod or coccus-shaped, rod and coccus-shaped, low GC content less than 55%, catalase negative, non-aerobic but aerotolerant, devoid of cytochromes, fastidious, acid-tolerant bacteria found in nutrient-rich environment like milk, meat, decomposing plant material and in the mammalian gastrointestinal tract. They are usually resistant to bacteriophages. They are known to have various attractive physiological properties and technological applications such as proteolytic activities, lactose and citrate fermentation, polysaccharide production, resistant to freezing and lyophilization, adhesion and colonization capacity to digestive mucosa, antimicrobial production. Lactic acid bacteria include the genera Lactobacillus, Aerococcus, Carnobacterium, Enterococcus, Lactococcus, Leuconostoc, Oinococcus, Pediococcus, Streptococcus, Tetraginococcus, Vagococcus, and Vacella. They are good biopreservative as their use in food extend the food shelf life and inhibit the growth of pathogenic and food spoiler microorganisms like bacteria, fungus, and yeast. This is because of their antagonistic properties as they compete for nutrients and also synthesize various inhibiting metabolites such as acetic acid, lactic acid, hydrogen peroxide, peptide bacteriocins or antimicrobial components similar to bacteriocin, also ethanol, diacetyl and carbon dioxide. Propionic acid bacteria convert lactate to propionate, acetate and carbon dioxide. They have been used in Swiss type cheeses and vitamin B12 production since long. They are gram positive, high GC content, catalase positive, non-motile, non-spore forming, pleomorphic, rod-shaped bacteria. Recently, propionic acid bacteria has found its role in food preservation. MicroGuard is a well-known propionic acid bacteria-based biopreservative sold by Danisco AS Denmark. It is a heat-treated ferment of Propionibacterium species without any living cells. The presence of diacetyl acetic, propionic and lactic acid and a heat-stable 700 Dalton peptide in microGARD are found to be responsible for associated inhibitory activities. MicroGARD inhibit most gram-negative bacteria such as Pseudomonas, Salmonella, and Yersinia, and some fungi and yeast. It is approved by Food and Drug Administration and can be used in yogurt, ricotta, refrigerated salad, sauces, fresh pasta, and fillings. Because of inhibitory activity of propionic acid and its salts against mold and bacillus, they are accepted as preservative for industrial use in bread manufacturing. Antagonistic behavior of yeast include competition for nutrients, production and tolerance of high concentrations of ethanol, pH changes in the medium as a result of organic acid production, synthesis of a large class of antimicrobial compounds which exhibit broad spectrum of activity against food spoilage microorganisms, plant, animal, and human pathogens. The yeast Debromyces hansenii has shown their antagonistic effect in citrus. Some of these species belong to Cryptococcus in apples and other fruits. The advantages of antagonistic features of yeast are they synthesize antimicrobial metabolites without producing any allergy-causing spores and mycotoxins. Nutrient requirements are simple and not very complex. They can be colonized on a dry surface for a long time. Use of yeast is economical as compared to the use of other chemical antimicrobial because substrate used for yeast growth are cheaper. They are also rich in large amounts of vitamins, minerals, and essential amino acids. Therefore, they are recommended for using in food and animal feed. Yeast is usually known to be killer yeast, which synthesizes either a killer toxin or mycosins. Mycosins are extracellular proteins or glycoproteins that kills the sensitive strains by disrupting cell membrane function. The most common mechanisms of killer toxins are interruption of cell division by blocking the DNA synthesis, inhibition of beta-1,3 glucan synthesis, and ion leakage through channels formed on cytoplasmic membranes. Here you can see some examples of killer yeast. Nowadays, the fruits after harvest are prevented from deterioration by using various kinds of commercial yeast. The commercial yeast product name as Aspire, which contains Candida oleifera, has been used in the US since 1996. Aspire is used to protect fruits such as citrus, apple, and pear from deterioration during storage by spray or immersion solution which contains a certain yeast concentration. Another commercial yeast product named as Yield Plus has been used in South Africa since 1997, protecting the various varieties of apples and the other fruits from deterioration caused by botrytis, penicillium, and mucor. The major content of Yield Plus is Cryptococcus albidus, antagonistic yeast. In Israel, the commercial yeast Mastinikovia fruticola was approved for sale in 2001 as a broad-use spectrum product shimmer for fruits such as strawberries, grapes, and sweet potatoes. Bacteriophages or phages are bacteria infecting viruses. The uses of phages against unwanted and harmful bacteria was proposed after a short time of their discovery. However, their uses in food was suggested recently by Struch et al. In the current scenario, the world is facing the challenge of increasing number of antibiotic resistant bacteria. The more of research on phage uses for killing such bacteria put a hope for sustainable solution of this challenge. Usually these research studies are more focused on the food safety, especially food pathogens such as Salmonella, Campylobacter, E. coli, and Listeria, which causes epidemics of disease. Recently, bacteriophages have allowed for use as antimicrobial agent in meat product in the USA. The phages also received a generally recognized as safe, that is GRAS status, as they are found lack of toxicity and other detrimental effects to human health. However, more studies over the phages are carried out to expand their use in food. A mixture of phages against Listeria monocytogenes was the first phage preparation to be commercially available for being used as biocontrol agent in food. The US Food and Drug Administration (FDA) on August 18, 2006 announced about the approval of using LMP102 phage preparation made from 6 purified phages with the trade name Listilt as an antimicrobial agent against Listeria monocytogenes on ready-to-eat meat and poultry products. The approved phage cocktail as an antimicrobial agent was found to be effective against 170 strains of Listeria monocytogenes. Nowadays, various phage preparations including specific for Listeria monocytogenes, E. coli, Salmonella, Enterica serotypes are available in the market for uses in food or as part of surface decontamination protocols. Most of the antagonistic microorganisms show their antagonistic behavior due to the synthesis of various kinds of metabolites affecting the growth of other microorganisms present in the vicinity. Their metabolites, along with some other metabolites obtained from plants and animals, can directly be used for the preservation of food. These metabolites include small peptides called as bacteriocins, organic acid like lactic acid, acetic acid, carbon dioxide, diacetyls, hydrogen peroxides, etc. The various criteria for an ideal biopreservatives are required to fulfill, such as low level toxicity, stable toward processing and storage, higher efficiency at low concentration, no detrimental effect on food, and economic viable. Lactic acid bacteria, through an oxidoreductive process, synthesizes organic acid as main end product. Some species like Lactobacillus, Lactococcus, Leuconostoc, Pedococcus, Streptococcus have been used as biopreservative in fermented food for long years. The organic acids particularly secure the food by reducing pH of the surrounding environment, generating a selective barrier against non-acidophiles. In addition, the organic acid also possesses antimicrobial effect on pathogens like E. coli, Staphylococcus aureus, Salmonella species, and Listeria monocytogenes in especially meat and dairy products, and also spoilage bacteria, yeast, and fungus in food products. Lactic acid kills the unwanted microbes by disrupting the cytoplasmic membrane and by interfering membrane potential and the intracellular pH. Acetic acid shows increased antifungal effect in the presence of propionic acid. Other organic acids have been found effective as antifungal biopreservatives. Lactic acid bacteria are deficient in catalase activity, therefore they produce hydrogen peroxide during their aerobic growth as a result of the action of flavoprotein oxidases or NADH peroxidases. Hydrogen peroxide has an antimicrobial effect on vegetative cells and spores of most of microorganisms through oxidative damage. Hydrogen peroxide kills or inhibits various pathogens such as Clostridium, Escherichia coli, Pseudomonas, and Staphylococcus. Diacetyl, having a buttery flavor, is a byproduct synthesized from pyruvate by lactic acid bacteria. Diacetyl is synthesized in higher amount by homofermentative lactic acid bacteria as compared to heterofermentic lactic acid bacteria. Lactic acid bacteria are highly resistant toward diacetyls. Diacetyls exhibit higher inhibitor effect on gram-negative bacteria, fungus, and yeast than gram-positive bacteria. The higher concentration of diacetyl required to inhibit the spoilage bacteria also affect the sensory property of the food. Therefore, the use of diacetyl producers as biopreservative is limited to food where diacetyl is an essential component of the food. Carbon dioxide is produced by heterofermentative lactic acid bacteria as a byproduct of sugar fermentation. It possesses antimicrobial effect. The presence of carbon dioxide creates anaerobic environmental condition, therefore inhibits the growth of aerobic bacteria. Gram-negative bacteria are usually more sensitive to carbon dioxide in modified atmosphere as compared to gram-positive bacteria. Also, lactic acid bacteria are found to be more resistant than obligate aerobic microorganisms. Many low molecular mass compounds synthesized by several lactic acid bacteria species have been identified with antimicrobial activities. They are thermostable, active in low pH, and can dissolve in acetone. Out of these compounds, only rotricillin and 2-pyrrolidone 5-carboxylic acid have been characterized, whereas others are not yet identified. However, the antimicrobial effect mechanisms need to be discovered yet. Bacteriocins are bacterially produced biologically active peptides having antimicrobial properties against other bacteria. They differ from antibiotics in being proteinaceous, which get digested rapidly in proteases in the human digestive tract. They are ribosomally synthesized peptides and therefore can be modified for enhancing their activity and spectra of action through protein engineering approaches. Most bacteriocins are thermostable cationic molecules with a size of up to 60 amino acid residues. Bacteriocins are synthesized by both gram-positive and gram-negative bacteria. The bacteriocin produced by gram-negative, named as microcin, are different in size, effect spectrum, and mechanism from bacteriocin produced by gram-positive bacteria. Many of bacteriocins are not found to be effective against gram-negative bacteria due to the presence of an outer membrane as permeability barrier. Mode of action. Many bacteriocin initially binds to the cell through electrostatic interaction with negatively charged phosphate groups on target cell membrane, followed by forming pores and killing the cells after causing proton motive force disruption, lethal damage, and autolysin activation to digest the cell wall. Many other bacteriocin may kill their target either through inhibition of protein synthesis, peptidoglycan formation, and spore generation, or interference with sodium and potassium ions transport. The characteristics of lactic acid bacteria bacteriocins which make them suitable candidate for use as food preservatives are: they are easily digestible by proteolytic enzymes of gastrointestinal tract due to its proteinous nature, non-toxic to lab animals, non-immunogenic to humans and animals, inactive against eukaryotic cells, economical, generally thermoresistance, broad bactericidal activity affecting most of the gram-positive bacteria, easy genetic manipulation for the synthesis of variety of natural peptide analogues as the genetic determinants are located in the plasmid. Here some factors in the food affect the production of bacteriocin negatively are: inappropriate physical conditions such as pH, temperature, and chemical composition of food like nutrients for growth or bacteriocin production. For example, niacin is 2 to 8 times more soluble at pH 2 than at pH 8. Spontaneous reduction in production capacity of strain, inactivation of the producing strain by phages, and antagonism effect of other microorganisms in food. Here some factors which effectiveness of bacteriocin activity in food negatively are: the emergence of bacteriocin resistance pathogens, inappropriate environmental conditions that may destabilize biological activity of bacteriocin, higher retention of the bacteriocin by food components like fat, inactivation of bacteriocin due to the presence of other food additives, degradation by non-specific proteolytic enzymes, activity dependent on narrow pH range, and partnering into polar or non-polar food components. Bacteriocins can be classified into 4 classes. Class 1, lentibiotics. This class comprises small, less than 5 kilodalton heat-stable peptides acting on membrane structures. These peptides differ from other bacteriocin in containing unusual thioether amino acid, lanthanidine, and beta-methyllanthanidine, and other modified amino acids such as dehydrated serine and threonine, which form as a result of post-translational modification. Class 1 is further subdivided into 2 subclasses. Class 1A consists of cationic and hydrophobic peptides having a flexible structure. Example is lysine. Class 1B globular and no or negative charge. Class 2 non-lentibiotics. This class comprises of variable molecular weight but usually small, less than 10 kilodalton heat stable non-lentibiotics linear peptide. This class was divided into 3 subclasses by virtue of having distinct N-terminal sequence. Class 2A having pyridosine like bacteriocins. Class 2B, these peptides have little or no activity, are lacking of leader peptide sequence. Class 2C, heat stable small peptides which are transported by leader peptides. Class 3, big peptides with a molecular weight over 30 kilodalton heat labile bacteriocins. And class 4 include large complex peptides require carbohydrates or lipid as moiety for activity. Microbial pigment possesses antioxidant vitamins property as suggested by several reports. Recently, the antimicrobial properties of these pigments have been reported. Monascus species synthesizes food coloring pigment, Monascus, which is the most widely known and used. This pigment has coloring ranges from yellow to orange, and this pigment has commonly used mainly in Far East for many years as a food colorant in several kinds of food items such as fish, cheese, beverages, wine, and even sausages. The antimicrobial effect of Monascus pigment varies from type of target microorganism to the type of pigments. Natamycin is the only broad-spectrum antifungal biopreservative approved for food and beverages produced by an actinomycete, Streptomyces natalensis. Since natamycin has a high affinity for the fungal cell membrane component ergosterol, this binds to the fungal cell membrane irreversibly, which makes membrane hyperpermeable, leading to the fast release of essential ions and peptides from the cells and ultimately cause cell lysis. Lactoperoxidase enzyme is naturally present in fresh raw milk, colostrum, saliva. 30 mg per liter of lactoperoxidase is present in bovine milk and its concentration is fairly constant throughout the lactation. However, its quantity is less in breast milk. This enzyme doesn't have the antibacterial effect its own, but in the presence of hydrogen peroxide and thiocyanate, this enzyme becomes effective and thus it can show antimicrobial activity, as you can see in this reaction also. While it shows a bactericidal effect against several gram-negative bacteria such as Campylobacter jejuni, E. coli, Listeria monocytogenes, Pseudomonas aeruginosa, Salmonella, Staphylococcus aureus, Yersinia enterocolitica, and also against coliform bacteria. It possesses bacteriostatic effect against some bacteria such as Streptococci and Lactobacilli. Lactoperoxidase system is usually more active on gram-negative bacteria than gram-positive bacteria. and this system has been used in raw milk as a protective component in the region where hygienic conditions are not good and during its transport. Various lactic acid bacteria, propionic acid bacteria, yeast, bacteriophages are the different microbial strains which can be directly used in preservation of the food. These strains behave in an antagonistic way and compete with other food spoilage and pathogenic microorganisms for nutrients. The various metabolites other than bacteriocins such as organic acids, hydrogen peroxide, carbon dioxide, diacetyls help these microbial strains in inhibiting the food spoilage and the pathogenic microbial growth. Some of the bacteriocin found active against antibiotic resistant pathogenic bacteria. Therefore, there is further need for isolation, synthesis of novel and effective bacteriocins.